Okay, we had discussed about cyclic voltammetry. The current responses of stationary electrode in an unstirred solution is excited by triangular wave voltage waveform, such as that shown in 25-23. In this example, the potential is first varied linearly from plus 0.8 volt to minus 0.15 volt versus an standard calomel electrode. When an extreme of 0.15 volt is reached, the scan direction is reversed and the potential is returned to its original value of plus 0.8 volt. The scan rate in either direction is 50 millivolts per second. This excitation signal is often repeated several times. The voltage extrema at which the reversal takes place in this case is 0.15 volt and plus 0.8 volt. आता ह्याच्यात आपण काय म्हटलं की आपण हा जो डायरेक्शन आहे सायक्लिक व्होल्टामेट्रिक एक्सायटेशन सिग्नल आहे तर तो प्लस 0.8 वोल्ट कडून आपण मायनस 0.15 वोल्ट कडे जाणार आहे आणि जसं तिथे काय होणार आहे इट विल बी गिविंग अ रिडक्शन of that analyte into the solution, and when we come to reverse the potential it will give us the oxidation of the analyte. Okay, the voltage extrema is at around minus 0.15 volt. Okay, so this is the way how we are going to operate and this is the example that is taken. So when we reverse the potential, that potential is called as switching potential. Okay, the range of switching potential chosen for given experiment is one in which a diffusion controlled oxidation or reduction of one or more analyte occur. So this is the way how we are going to choose the switching potential. What is switching potential? It is this value that we are going to talk about. Okay, here it is shown what we saw in the upper figure. The same figure is drawn over here. So what we are going to do, we are going to have this particular value of switching potential considered. When we go from A to B to C, D, E, F, how we are going to show or observe what are the effects of potential applied will take place to the electrode that are indicated in this figure. So what is A? A is the starting potential and F is the switching potential. Okay. And when we go with the directional change in the potential magnitude in a triangular wave, what current changes takes place that are plotted as cyclic voltammo graph. Okay, I hope you understood what is meant by switching potential. F is the switching potential and what is the magnitude of it? Minus 0.15 volts. Okay, and that will depend upon what we are going to have the experimental condition. Experimental condition कशावर dependent आहेत त्या 0.15 volt कुठून आला तर that will depend upon what we are going to have the analyte in the experiment selected. What is the oxidation and reduction range of that analyte will be deciding what we are going to have the particular magnitude of switching potential selected. The direction of initial scan may be either a negative as shown or positive depending on the composition of the sample. Okay, what is meant by negative and positive that we talked in the previous lecture. A scan in the direction of more negative potential is termed a forward scan. So when we go with a more negative potential, it will be called as forward scan, and one in the opposite direction is called as reverse scan. So generally cycle time range from 1 milliseconds or less to 100 seconds or more. In this example, the cycle time is 40 seconds. In this case, what we are going to have the time selected, time will be 40 seconds for which we are going to complete this cycle. So altogether, we are going to have this particular triangular wave that will be applied for 40 seconds. Now, in this figure 25-24b, so that is what we are going to have this figure. So this figure indicates what we are going to have the current responses recorded with respect to how we are going to apply the potential. Okay. So this current response when a solution that is 6 millimolar in K is K3FeCN6 and 1 molar in KNO3. So this is our analyte and this is the supporting electrolyte. So this is subjected to cyclic excitation signal shown in figure 25-23 and 25-24a. So these two figures indicates what we are going to have the cyclic voltammo ground and how we are going to work with that particular working electrode. The working electrode was carefully polished stationary platinum electrode, and the reference electrode was a standard calomel electrode. So as I told you, voltammetry will talk about working electrode as a stationary electrode. So stationary electrode selected over here is stationary platinum electrode. Okay. At the initial potential of plus 0.8 volt, a tiny anodic current is observed, which immediately decreases to zero as the scan is continued. So this initial negative current arises from the oxidation of water to give oxygen. At more positive potential, the current rapidly increases and becomes quite large of about plus 0.9 volt. So in the initial way, we are going with an plus 0.8 volts. So that we have seen, we started with plus 0.8 volt, and the tiny anodic current is observed, which immediately decreases to zero. So this is what we say it about. Hazo part hai, it indicates we are going below zero cha khalti anodic current, and this is cathodic current. Positive direction will be indicated with cathodic current, and negative will be indicated with the anodic current. So when we go with initial change in the potential, we observe some drop in the current magnitude from zero to negative direction, but that is immediately increased to the value. हे सगळं काय आहे खाली गेलेला anodic current we are calling it as. Okay, so this will take place in that solution when we go with a plus 0.8 volt scan. So again it comes to zero as the scan is continued. So this remains at zero unless you come to the Faradic process taking place at the working electrode. This current is called as, yesterday we talked about the part which is called as condenser, charging, capacitive or residual current. Okay, so initially whatever current we get, that will be called as residual current, and that remains to zero until and unless Faradic process starts. The initial negative current arises from the oxidation of water to give oxygen. So this contribution is done because of the what we are going to consider it as negative value of current, that will be oxidation of water to give oxygen. At more positive potential, this current rapidly increases and becomes quite large at about plus 0.9 volts. So when we start with the scan, as we are going with to the plus 0.9 volt, it can lead to give a large value of negative current, but we are not going to the larger value. We are not going at this side of the potential. We are going on the other side. We are decreasing the potential to the negative scan. All right. We are not going to the plus 0.9 volt. We are coming to the lower value, plus 0.8, plus 0.6, plus 0.4. Like that, we are coming till minus 0.15 volt. All right. So this is the answer to the question that why we are having decrease initially. Decrease is due to what we consider, it is due to oxidation of water to give oxygen at what we are going to have more positive potential. If we are going in the more positive direction, जर का आपण इट जो उलटा घेतला असता तर आपल्याला हा आणखी anodic curve मिळाला असता, anodic current मिळाला असता. So that we are not doing it. We are going with a negative scan or cathodic scan. So no current is observed between a potential of plus 0.7 volt to plus 0.4 volt. हा जो रेंज आहे इथे खाली दिसतं तुम्हाला काय, प्लस 0.7 वोल्ट पासून आपल्याला प्लस 0.4 वोल्ट पर्यंत आपल्याला नो करंट. So there will be zero current, that is no current in the system is recorded till what we are going to have the point which is called as plus 0.4 volt, because no reducible or oxidizable species is present in this potential range. In this range, there will be no component that can get oxidized or reduced that is available to the electrode surface. So potential range applied will be not giving any current or non-Faradic processes that will take place. When the potential becomes less positive, then approximately plus 0.4 volt, ह्याच्या आणखीन पुढे जाऊन आपण आणखीन plus 0.4 volt less positive कडे जातोय, plus 0.4 volt कडनं plus 0.2 volt कडे जातोय. So that will give when as you are going down or as you are going to the negative scan, because of reduction of hexacyanoferrate, that is what we are going to have reduction indicates K3 FeCN6 is going to K4 FeCN6. Plus 3 step will go to plus 2 step by reduction of Fe plus 3 to plus 2. So we are going to have this K3 FeCN6, which we are going to have initially present in the solution, that will go to K4 FeCN6 as you start lowering down the value of potential to the negative scan or less positive scan. Alright, understood? So a rapid increase in the current occurs in the region of B to D. So due to reduction, as the process starts of reducing K3FeCN6, it will have an increase in the current, and that will be observed at point B. It starts increase in the current from B point, and to D it increases. So you have this particular value of current that will be rise, like an polarograph. How we have the polarograph, till you have all components which in the vicinity of the electrode, that will get reduced. You are going to increase the potential magnitude. So at the surface, as the surface concentration of FeCN6 3 minus becomes smaller and smaller, it will rise the current magnitude. The current at the peak is made up of two components. One is initial current surge required to adjust the surface concentration of the reactant. So in the initial, when the initial current rate surge means what? Whatever is the required magnitude of surge was there, or thrust was there, that we have talked about on the surface of electrode, because of the concentration gradient form, you are going to have first initial uh, uh, peak will be contributing this particular magnitude of current. All right. So we are going to have this particular value. One is the initial current surge to adjust the surface concentration of the reactant to its equilibrium concentration as given by Nernst equation. And the second is the normal diffusion control current. So this, as we say in case of polarography, same thing is happening in case of voltammetry. At this point, you are going to have limiting value. You are going to achieve the equilibrium between what is the concentration at the electrode surface and what is the diffusion rate of the analyte from the bulb to the electrode surface. So this too will contribute the peak current. All right, this magnitude of current. So as you start increasing the potential, you are going to reach the value of current maxima, and that will be indicated in this value. Okay. The first current then decays rapidly. Okay. Now point D to F. When we start with the point, what we are going to have the lowering or going to positive direction of potential. So you are observing here where we are over here. We are going to up to minus 0.15 volt, and we come to a point whereby you are going towards the value of negative scan. So negative direction scan will be stopped over here. So as you go from point D to F, you come up to minus 0.15 volt. So this is the value of minus 0.15 volt where you reverse the potential. So current at the peak, after that it decays, it starts decreasing. As you start coming to negative scan, you come to the point where you are going to have lowering of current. All right. So the first current then rapidly at what we are going to have the decreasing magnitude of current that will be observed. Then how we are going with the point at which you reach minus 0.15 volt, the scan direction is switched. So what is meant by that? We are going to decrease the potential range from negative to the positive value. So you go towards the positive scan. The current, however, continues to be cathodic. Now you can observe here in this peak, still you are getting a cathodic current. Cathodic current means what? Above zero value, it will be having a cathodic current. So it is not coming down. It is not an symmetric peak, right? So it is not coming down and becoming zero, like what we consider over here. As you go down, you should have an anodic current. So as you are going towards from F value, where you are having switching potential, still the current remains cathodic. So here it comes down, but it still remains cathodic, and that is observed and plotted in case of cyclic voltammetry. The current, however, continues to be cathodic even though the scan is towards more positive potential because the potential are still negative enough to cause reduction of Fe6. Now, why we are getting this contribution? We say that this current we consider it is cathodic because potential is in still negative range. Coming to positive value will come to reach for oxidation range of the analyte. So what we say, the process of reduction continues. Still you have the negative scan done or switched the potential from minus 0.15 volt to the positive direction. So you are getting still reduction of K3FeCN6 to K4FeCN6 at this range and contributing the part which will be called as cathodic current. Alright. So as the potential shifts in the positive direction, eventually reduction of FeCN6 no longer occur and the current goes to 0 and then becomes anodic. So here we are going to have the decrease in the value of current to 0. So at this point it becomes 0 and then it becomes anodic. So here you say it will be reverse. ह्याच्या उलटा प्लॉट आपल्याला इथे मिळतोय. हा काय होता कॅथोडिक करंट होता by reduction of K3FeCN6. Now we come to the point where from this you will observe that this will be reversal of the oxidation will take place. So from the 0 point you are again having the oxidation of K4FeCN6 to K3FeCN6 that will be observed from this point of the potential. So what is the range of potential? It will be at around 0.2 volt. So as we said, it will be having the anodic current resulted from reduction and has accumulated near the surface during the forward scan. The anodic current peaks and then the decrease as the accumulated FeCN6 is used up by the anodic reaction. So what we are going to have, K4FeCN6 is going to get reduced to K3FeCN6 and that will decrease till you have the complete reduction at the electrode surface. So anodic reaction will stop as you are having no further reducible analyte is available at the electrode surface. So K4FeCN6 is consumed and then you are going to have anodic current stops to grow in magnitude as the anodic current. Alright. So this is how we are going to have this particular part of what we consider cyclic voltammograph of K3FeCN6 and K4FeCN6. Okay. Is there any doubt? If you are having any doubt, then you can ask me in the discussion session here, and then we will discuss about the cyclic voltammograph of how we are going to carry out for parathion. Okay. Now, how we are going to go with the reversible scan? We can have an irreversible process that can be discussed over here. So we are having here a reversible process. In this case, we are going to have an irreversible scan along with an reversible scan. All right. So actually, when we talk about parathion is considered as the component that will be insecticide. Cyclic voltammogram of insecticide parathion is indicated over here. So parathion gives us a plot as you go with an potential scan. How we are going to get one irreversible process and one reversible process that can encounter with a different decomposition potential. All right. So before we start with actual parathion molecule with an reversible and irreversible processes, let us talk about how we are going to have the important variables in cyclic voltammetry that are considered over here. So what we talked about EPA was potential, peak potential. So in EPA is the peak potential magnitude, EPA here, and EPC. So these particular values that can be related with what are number of moles of the substance. So how we start with the analysis, we come to the part of recording EPC and EPA, that are anodic peak potential and cathodic peak potential. IPC denotes cathodic peak current and IPA corresponds to anodic peak current. The definition and measurement of these parameters are illustrated in figure 25-24 as we discussed already for a reversible electrode reaction. So this is reversible. what we talk K3 efficiency and K4 efficiency, that Fe+2, Fe+3 reaction is taking place. So for a reversible electrode reaction, anodic and cathodic peak currents are approximately equal in absolute value, but opposite in direction. So you can say that peak potential and peak current are actually in the value of same value, but they are in opposite direction. So making with an opposite sign. So for reversible electrode reaction at 25 degrees centigrade, the difference in peak potential delta EP is expected to be delta EP is equal to EPA minus EPC is equal to 0.0592 upon N. So for reversible reaction, we can write this equation. Where from you are getting this? Actually, when we substitute the value of potential often, we can get the value in the opposite direction. So it cancels out of all other parameters in Nernst equation, and that gives us a simplified way of 0.0592 N. And we can find out what is delta EP. Delta EP is what value? It is the difference between these two peak potential. So this gap is there, that delta EP can correspond to 0.0592 upon N. Alright. So where N is the number of electrons involved in the half reaction. So obviously for Fe+3, Fe+2, N will be 1, and therefore delta EP will be directly equal to 0.059 or 59 millivolts. So irreversibility of because of slow electron transfer kinetics results in delta EP exceeding the expected value. So when we have this particular irreversibility as shown over here, or when we talk about irreversible reaction, the opposite component is absent, opposite sign is absent. So delta EP will be exceeded the value of expected value. Although an electron transfer reaction may appear reversible at a slow sweep rate, increasing the sweep rate may lead to increasing value of delta EP. This indicates what? We say that this is the, we are talking about sweep rate. That is the frequency at which we are scanning the potential. So this is very important part. Sweep rate indicates what, at what rate you are changing the potential magnitude at the electrode. So we selected in this case for K3 efficiency 40 milliseconds. So that was a sweep rate. Sweep rate indicates it is the frequency at which you are continuing the cycle. For 40 milliseconds, we are running that triangular wave of potential. So same way, repetitive potential scan can be made with a constant sweep rate. If we slow down the sweep rate, it increases. So increasing the sweep rate may lead to increasing value of delta EP. Slowing down will decrease the delta EP. Is sure sign of irreversible. Hence, to detect slow electron transfer kinetics and to obtain rate constant, delta AP is measured for different sweep rates. See, quantitative information is obtained from Randall-Servik equation, which is at 25 degrees centigrade. IP is equal to 2.686 into 10 raised to 5 n raised to 3 by 2 a c d raised to half nu raised to half. Very important equation because we are using this equation for solving problems when we talk about quantitative analysis. So by using cyclic voltammetry, how we go with the quantitative analysis, we will see how we can have the effect. But what is the actual equation relates with? IP is the peak current, A is the electrode area. So when we select a solid electrode, stationary electrode, the A denotes its area in centimeters square. D is diffusion coefficient in centimeters square per second. C is the concentration and nu is the scan rate. So cyclic voltammetry offers a way of determining diffusion coefficient if the concentration, electrode area and scan rates are known. So this equation actually, when we take simple same electrode system, this all other parameters remains same. And what will be varied? We can change the potential nu as the scan rate, volts per second, and we can have a direct relation obtained between IP and scan rate. Increase in current will be increased with the increase in the scan rate. So half square root of scan rate is directly proportional to peak current. Alright. So this equation we are using, and we are going to see one to two problems based on this equation. So I hope you understood and followed everything as we discussed over here. We will see in the next lecture cyclic voltammogram of insecticide parathion. Okay, stop here.